All right, guys, so to begin with, a little bit of review of things we've already talked about. The cell cycle, the life of the cell. This diagram depicts what it looks like. Most of the time, a cell is not dividing. Most of the time, it's in a part of the cell cycle called interphase. That means between cell divisions. Interphase is made up of three parts: G1, S, and G2. Right in here, you got brand new cells. And during G1, they grow, and they do their job, whatever their job is. G1's a pretty long stage. The length of the arrows represents time. After G1, though, they go into what's called the S, or synthesis phase. What gets synthesized? This is when the DNA is copied. DNA replication happens here. So even though this isn't cell division, these cells are getting ready to divide. The DNA is copied, replicated. Eventually, the cell goes into G2, the last part of interphase. In that part of the cell cycle, the cell continues to do whatever it's supposed to be doing, its job. It makes some extra copies of its organelles, and it also begins to build these microtubules that it's going to use to do mitosis. Finally, the cell comes out of interphase, and it goes into what's sometimes called the M phase. And that's really made of two parts. Mitosis is what always comes after mitosis that's not written down up here. Cytokinesis. Okay? Because remember, mitosis is not really cell division. Mitosis is the division of what? The DNA. Division of the DNA, the nucleus. And then cytokinesis comes right after that and divides the cell in half. So the M phase is made of two parts: mitosis, cytokinesis. That's the cell cycle. The cell continues to do its job. It makes some extra organelles. It makes some microtubules. We talked also about this slide already, how that cell division happens fast. You make up to 2 trillion new cells every day. A lot of those are blood cells, skin cells, cells in the linings of your stomach. And that one of the reasons cell division happens is that you have to replace a lot of old cells. When you grow, cell division makes you grow. And also repair. There's one thing that's not up there that probably should be. We also asked a question yesterday, I think it was, can bacteria, can they do mitosis? No, because they don't have what? They don't have a nucleus. Mitosis is really division of the nucleus. But they do something really similar. And it's called binary fission, or up here it says prokaryotic fission. And it looks sort of like, I'll go to a picture here. That's what binary fission looks like. Bacteria have one chromosome. First of all, they copy that chromosome. There's 2 of them here. And then that cell grows, pulls those 2 chromosomes apart, and eventually splits down the middle. This is a form of what kind of reproduction? Asexual. In some bacteria, they do it as fast as about 30 minutes, the whole thing. So it's fast.
Why bacterial viruses?
That's why one bacteria in a day can become more than 1 million, if the conditions are just right. We talked about some terms too, and we need to make sure we know what these terms are. Yeah. Remember that most of the time, your DNA is not in what form? Most of the time you don't have chromosomes. Most of the time your DNA is unwound, sort of like a plate of spaghetti noodles. It's all spread out. When it's in that form, it's called chromatin. This is when it's working. This is when it's used to make what? What's DNA used to make? Proteins. This is when it's doing that. But just before cell division, well, before mitosis even starts, what happens to that chromatin? What has to happen to the DNA before mitosis starts? Before that, it has to get copied. So first of all, it gets copied, and then it does what? Then it winds up. Because remember, in mitosis, you're trying to split the DNA into 2 equal chunks, right? And if it's not equal, we talked about all the problems yesterday that could happen, right? It needs to be exactly equal. So we normally think of a chromosome as this X-shaped thing. But yesterday I told you that's really what? It's really 2. And that's why we call this egg-shaped thing a bivalent chromosome. It's really 2 copies of a chromosome, connected in the middle by what little structure? A centromere. Centro just means center. And each half of this is called what? Chromatid, or sometimes we call them sister chromatids. And we know that during mitosis, what's going to happen to those 2 pieces? They're going to split. One half's going to go into one cell, one half's going to go into another. We also gave you some names for a couple of kinds of cells yesterday. We talked about somatic cells. We said those are just what? Yeah, your normal body cells, not related to sex. These are like skin, blood, bone, cells not related to reproduction. Somatic cells. Well, hair is actually just protein, but the cells that make the hair. We also mentioned germ cells yesterday. These are cells found only in certain parts of your body, in your ovaries or your testes. And these are the cells that can do what kind of cell division? These germ cells are the only ones in your body that can do meiosis. The main job of meiosis is to create what? What does meiosis make for you? It makes sex cells. It makes gametes. It makes sperm. It makes eggs. So these germ cells do meiosis and become gametes, or sex cells, sperm, and eggs. So those are all some important terms to know.
I'm informed with all of those.
Yeah, and we'll get to that. When we get to meiosis, we'll talk a lot more about that. Here's some other terms that we've already talked about that I want to make sure you know. We said in you ladies, how many kinds of chromosomes do you have in each one of your cells? 23. But you have how many of each of those 23? You have 2 of each. Those chromosomes that are pairs are called homologous chromosomes. So you got 1 of each of the 23 from each of your 2 parents, right? You got a chromosome number 1 from dad, you got a chromosome number 1 from mom. They together are called a homologous pair. Now, homo usually means what? Same. Are these chromosomes in a pair exactly the same? Is the chromosome you got from dad exactly like the one you got from mom?
No.
There's no way, right? But what is the same about them? Let's say this one you got from mom, and this one you got from dad. If the first gene on that has something to do with how tall you're going to be, what's the first gene on this one have to do with? How tall you're going to be. One may say you're going to be a little bitty shrimp, and one may say you're going to be a giant. You are a mental giant, right? But they have the same genes, but they may have different versions of those genes on there. This one may say you're going to have brown hair. This one may say you're going to have blonde. But they have the same. They control the same traits. The order of the genes, the order of the traits is the same, but they may have different versions of them. Those are homologous chromosomes. You ladies have 23 pairs of homologous chromosomes. What about us guys? How many pairs do we have? We have 22 pairs, and then we have 2 kind of oddballs, right? What are the 2 oddballs? X and Y. We have 22 pairs of homologous chromosomes, and then we have an X and a Y that aren't a pair, because they don't have the same way. What do we say homologous chromosomes have that are the same? Same genes. X and Y don't. They're not a pair. And that's why us guys sometimes end up with what? Weird disorders that women don't usually get. Because those 2, if we get a mutation in either one of those, the X or the Y, what happens? It shows up. Right? We have issues. So a couple of words related to that. This is an important word, diploid. Di means 2. It means 2, right? Your cells are diploid if they have what? What does diploid mean? Having 2 of each what? Kind of chromosome. And all your cells are diploid except for which ones? The sex cells, the gametes. Notice that sometimes instead of saying the word diploid, we may just write 2N. And that just means 2 of each, right? It's like in algebra you say 2x. 2 of each. Your somatic cells, your normal body cells, are all diploid. On the other hand, your gametes, your sperm or your eggs, they're haploid. They only have what? One of each kind of chromosome. And that would be how many? How many chromosomes should be in a normal sperm or a normal egg? 23. Half the normal number. That's what haploid means. So again, gametes are haploid. Other body cells are somatic cells, and they're diploid. Now, you should also know that during fertilization, a haploid sperm gets together with a haploid egg, right? Haploid plus haploid gives you what? Diploid. And that first cell that you start with after fertilization, that first cell that became you is called what? Your zygote. Zygotes are always, in us, going to be diploid, right? 2 of each. So what do we say happens if a zygote is formed that has the wrong number of chromosomes? Almost always dies. If it doesn't die, it gets some weird genetic disorder, right? Yeah.
A star from birth or something like that. Like my little brother, he has like a brown spot in his eye. How's that? Your little brother?
I'd have to know more about what's going on there. It's like in the white of his eye?
Yeah, it's a birthmark. It's like in his pupil, like not in his pupil, but like his eye's green, and then in one little square spot is brown.
So it's in the iris, the colored part, or it's in the white part? I'm not sure there.
It's like a brown little spot.
Interesting. I don't know. All right, couple more terms. Yes. Go ahead.
What's the color of your eyes? Like I have like 2 different colors in my eyes. Oh. Yeah.
Yeah, and there are a lot of people like that. I mean, nobody really has straight up blue eyes. Nobody really has straight up brown eyes. If you really look close, there's splotches of different colors in there, right? And that's just the way it works. Yeah.
Eyes like change color.
Yeah.
Yeah, because I don't want to get too far off track with eye color right now. Need you guys to focus. Eye color is about a pigment. The same pigment that makes your skin color makes your eye color. It's called melanin. And if you have more of it at a certain time, your eyes are darker. If you have less of it, your eyes are lighter. And at different times, depending on what's going on in your body, you may make more or less. So your eye colors can change.
So
That's as far as I want to go with that right now. Let's talk about the rest of these terms. Autosomes are chromosomes in your cells that have nothing to do with what? Sex. And how many of those do you have in every cell? Except your sperm or eggs.
Wait, nothing.
Just wait. We're gonna do this right now. We have 44 autosomes in every cell, except sperm and eggs. And then we have 2 chromosomes that determine your sex, and those are your sex chromosomes. And as we said before, you're ladies because you have what? You have an X and an X. That's what makes you be a lady genetically. You guys, on the other hand, you're guys because you have an X and a little bitty chromosome called the Y. And essentially, the only genes on that Y are genes that make you be a guy, right? Not really it. Can people live without Y chromosomes?
Yep.
Yeah. Half the people in here are living just fine without one. But they're the chromosomes that make us be guys. All right, let's go a little further here. This is the last slide I think we looked at. We talked about similarities and differences between mitosis and meiosis. This column over here is for mitosis. This column over here is for meiosis. Mitosis is our main kind of cell division. Mitosis happens everywhere in your body, head to toe. It essentially takes one cell and makes how many? Mitosis takes one, and you end up with two. And how are those two that you make related to the one you started with? There, just like. We might associate the word cloning with mitosis. Because that's really what it does. Mitosis takes one cell and makes two that are just like it. It just rolls in the body, lots of them. It makes new somatic cells, new body cells. It replaces cells that have died. Mitosis helps you grow. Mitosis repairs damaged places, like a cut, a break. It fills in those holes. And mitosis is how most things that do asexual reproduction, it's how they do it. Like a little amoeba splitting into two amoebas. That's mitosis. So those are all things related to mitosis. Do amoebas have what? Yeah, they're eukaryotes. On the other hand, this right-hand column are things related to meiosis. Meiosis is a specialized kind of cell division. It only happens, here it says the word gonad, but that means in your ovaries, if you're a woman, or in your testes, if you're a guy. It's the only place in your body where this happens.
Why would you just prefer one or one?
Well, some of it happens before birth. Some of it happens after, though. It's not finished yet. And when we get into meiosis next week, you'll understand. Next week after next. But meiosis' job is to make sperm and eggs. This way. Meiosis' job is to make sperm and eggs. It creates haploid cells from those cells we talked about at the beginning of class today, the germ cells. These cells are diploid. This is like a normal cell. But it has the ability to do meiosis. Essentially, meiosis does what to the number of chromosomes in a cell? You start off with cells of 46. When meiosis is over, what do you got? Cells with 23. It halves the number. And that's important to note. Meiosis halves the chromosome number. In meiosis, you start with one cell, but what do you end up with? You end up with 4. Because each one of those cells divides twice. This cell divides in half, and then these two divide in half again. So you end up with 4 cells. And these 4 cells, how do they relate to the one we started with up here? They're different. They're genetically different, and they have what chromosome number compared to the cell we started with?
23.
Half. If this one started with 46, these 4 each have 23. Now you might be thinking the math doesn't add up. Right? The math doesn't add up. But what happened to the chromosomes in that cell we started with before all this started? It copied. So really, that cell up at the top didn't start with 46. It started with what? 92. Okay. And what's 23 times 4? 92. So the math does add up. It's just that this cell normally has 46 chromosomes. But just before meiosis starts, those 46 get copied. So it really becomes 92. The cells divide twice each, and we end up with 23. These cells grow up to be sperm or eggs. That's how we make gametes. All right, now that's kind of stuff we've already talked about. What we're going to do for the rest of the time today is go into some stuff we haven't talked about. I'm going to come back to this stuff about cancer, but what I want to focus on today here first is mitosis and meiosis. Mitosis mostly. And I want to talk about the different parts of mitosis. Mitosis, we're going to talk about 4 stages of mitosis. Some books are now dividing it into 5. We're going to divide it into 4. The first stage that we're going to talk about is prophase. Prophase. And we can use a little acronym to help us remember the order of the different parts of mitosis. PMAT. That's the order. PMAT. First stage is prophase. And before we start on prophase, let's look at a picture before we get started. This is what a normal cell looks like when it's not dividing, which means it's in what phase of the cell cycle? Interphase. Is interphase part of mitosis? It's what happens before mitosis, right? So this is a cell that's not in mitosis, it's in interphase. Notice that it's got a cell membrane. It's got a, what's this right here? It's a nuclear envelope, nuclear membrane. And then the DNA in here is kind of all unwound, sort of like spaghetti noodles. And it's got a little nucleolus in there. That's a normal non-dividing cell. But then we hit prophase. And there's a list of things up here that happen during prophase. First of all, the chromosomes form, which means the chromatin did what? Where's the chromosome come from? It comes from chromatin that did what? No. That winds up. So let's say this bow tie here is chromatin. DNA in its unwound form. When it's time for mitosis to start, that DNA winds itself up. And that's what a chromosome is. It's DNA in its unwound form. Chromosome is DNA in its wound up form. Chromatin is DNA in its unwound form. No. Here's the thing. The DNA is about to get divided, right? That's the purpose of mitosis. If it's all unwound, is it going to be very easy to divide evenly? No. The purpose of a chromosome is to act sort of like a spool of thread, to organize that thread, in this case the DNA, in a way that it's easy to split. Wind it up. That's what happens first. Chromosomes wind up. DNA winds up to make chromosomes. Also, the little things called the spindle fibers form. We'll talk more about spindle fibers in a second. I'll show you a picture. The nucleolus, that little dark spot in the nucleus where ribosomes are made, it goes away, it disintegrates. We're going to include what's listed here as prometaphase. We're going to include that as prophase. So something else starts to happen. The nuclear envelope starts to fragment, it starts to break down. And the final thing that happens, the chromosomes connect up to the little spindle fibers. There's like 5 things that happen in prophase that are important. Let me go back through them again. These are things you need to have listed. The chromosomes form. The spindles form. The nucleolus, it goes away. The nuclear membrane starts to break down, it starts to fragment. And the chromosomes connect up to the little spindle fibers. 5 main things that happen during what we're going to call prophase. Chromosomes form, spindle fibers form, nucleolus breaks down, nuclear envelope breaks down, and chromosomes connect up to the spindle fibers. So we're going to look at a picture of this in a second so you get an idea. Because one thing you're going to have to recognize from pictures is what stage you're looking at. Tomorrow we're going to do a lab where we're going to take some cells from an onion, the plant, and we're going to look at, they're going to be stained so you can see the DNA in there, and you're going to have to be able to identify what stage of mitosis they're in. So we'll get to that tomorrow. That's tomorrow. Keep it here for just a minute so you can finish writing it down. 5 main things again though. The chromatin winds up and becomes chromosomes. That's number 1. Number 2, the spindle fibers form. Number 3, the nucleolus disappears. 4, the nuclear envelope, the nuclear membrane breaks down. That makes sense because what are we trying to do to the nucleus? Split it. You got to break that barrier if you're going to split it. And then lastly, the chromosomes attach up to these little rope-like structures called spindle fibers.
What was number 1 again?
Chromosomes form. Chromatin winds up to make chromosomes. So I'll talk about these on the next slide, if you don't have it all down. Here's a picture of what the cell might look like at the beginning of prophase. What's already gone in this picture that was there a minute ago? Nuclear envelope is gone. Notice that the DNA is now in that famous X shape, right? Chromosomes are now there. The chromatin wound up and made chromosomes. Notice that over here, these are the little rope-like structures I was talking about. These are spindles. These spindles are beginning to form. It almost looks like a spider web that's forming, right? That's the beginning of prophase. If we go a little bit further, this is what I'm going to call the end of prophase. By the end, notice, again, the nuclear envelope is gone. The DNA is all wound up in the form of chromosomes. Now we've got this spider web-like network of spindle fibers, little pieces of protein. And also notice the little nucleolus is gone. All the chromosomes are attached up to one of those little rope-like spindles. That's kind of where prophase ends for us. All right, remember our acronym, PMAT, P-M-A-T. So the next phase is the M part, and that's metaphase. Anybody know what meta means?
So metaphase is number 2.
Metaphase is number 2.
Meditate.
Anybody know what meta, just M-E-T-A means?
Middle?
It means middle. Because what's about to happen is these little things, remember, these little spindles are like ropes. And their job is to do what? Pull the chromosomes where they need to go. Again, think of them like ropes. So the first thing that happens in metaphase, the chromosomes get pulled right into the middle of the cell. They get lined up, like single file. Chromosomes all get lined up in the middle of the cell. Metaphase. And that's really all I need you to know at this point about metaphase. The chromosomes are pulled to the middle of the cell, lined up. And in a picture, it might look like this. You see how all the chromosomes have been lined up in the middle. Now, is this a human cell we're looking at a picture of here?
Probably not.
Probably not.
No, because if it was, how many chromosomes would be lined up right there?
It's 3.
How many chromosomes do you have in a normal somatic cell? 46. The reason they only draw 4 is it would be hard to draw 46 in a line, right? It's probably just some kind of cell they made up. But there would be 46 in a line if this was a human cell. It's not. All right, so that's metaphase. The little ropes, the little spindles, pull the chromosomes to the middle, line them up. Again, go back to our acronym. Prophase, metaphase, and then we get to the A part, and that's called anaphase. That's step number 3, anaphase. So in anaphase, essentially those little ropes that we were talking about, those spindles start to shorten. The ropes on one side pull toward the right, the ones on the other side pull toward the left. What's about to break? Those X-shaped chromosomes are about to break in half. And they're going to separate at the centromere. So what this says right here, the spindle fibers help to pull apart the sister chromatids. Remember, our chromosomes look like that. What's going to happen during anaphase is that this part's going to break. And one of those chromatids is going to get pulled this way, and the other one is going to get pulled this way.
What's the phase called?
What's what?
The phase called?
Anaphase.
Number 3.
Number 3. What do we know, though, about this half and this half, if they were together at one point in time? How are they related? Hey, let's go.
This is for James.
Okay, that's James. Thank you, man. What do we know about the two halves of a bivalent chromosome? How are they related? They're exactly the same. Just what are we trying to do in mitosis? Make 2 cells that are just alike, right? That's what we're trying to do. We're trying to copy. So during anaphase, what you need to know is this sentence right here. The spindle fibers pull apart the sister chromatids. One of those chromatids starts getting pulled toward one side of the cell. The other one gets pulled toward the other side. That's anaphase. And let's look at a picture of how that might look. Here's what a cell in anaphase looks like. These chromosomes were all together a second ago. Now these little ropes have shortened. One of each chromatid is getting pulled toward this side. One of each chromatin is getting pulled toward that side. This set of chromosomes is related how to that set of chromosomes? They're exactly the same. Because remember, mitosis is like a copy machine. It's trying to make two identical sets of DNA. Split the DNA into two identical sets. Back to our acronym. Prophase, metaphase, anaphase, and the last stage of mitosis was called telophase. Some people say it telophase. I usually say telophase. So let's go back a second. During telophase, the main thing that happens is that the chromosomes make it to opposite sides of the cell. Chromosomes make it to opposite sides. So you've got one set of chromosomes on the right, one set on the left. From there on out, telophase is pretty much the opposite of prophase. So let's think about that for a second. Remember during prophase, what happened to the DNA? No, that was before. What happened to the DNA during prophase, the first stage of mitosis? It wound up to make a chromosome, right? Well, notice that in telophase, the opposite happens. The chromosomes unwind and go back to being what? What's an unwound chromosome? Unwound DNA called? Chromatin. So the chromosomes unwind, they go back to being chromatin. What happened to the nuclear membrane during prophase? During prophase, it disintegrated, it went away. So in telophase, a new nuclear membrane forms around each set of chromosomes. So for a minute at least, you're going to have one cell with what? Two nuclei. During telophase, you're going to have one cell that now has two nuclei in there. Also remember that during mitosis, during prophase rather of mitosis, what happened with the little spindles? They got made. During telophase, they get what? They get broken down. They get disintegrated. Mm-hmm.
How can the nucleus form around the chromosomes if they turn back to chromatin?
Well, this is all going on at the same time, so we could say around each set of chromatin, that'd be okay, but it's all happening at about the same time. Yeah.
I'm sorry, what was the role of prophase again?
We'll come back to that in a minute. We'll finish telophase first. Also remember that during prophase, the nucleolus got broken down, it went away. During telophase, a new nucleolus shows up in each new nucleus. So let's go back to the pictures and we'll summarize what happens in each stage from the pictures. Here's telophase. Notice at the end of telophase, we have still just one cell. But that one cell has what inside it? Two. What are these? Two nuclei. At the end of telophase, we have one cell with two nuclei. Each one of these nuclei has a membrane around it, a nuclear membrane. Each one now has a little nucleolus in it. And the DNA is unwinding, and it's going from a chromosome back to chromatin. So let's go back to the pictures here for a second and talk through them again. Here's interphase, not part of mitosis. It's a normal cell. In interphase, cells are doing their job. Whatever they're supposed to do, they're not dividing. An interphase cell should have a nuclear membrane. It should have a nucleolus. And it should have DNA that's unwound and not in the form of chromosomes. It's chromatin. It's interphase. But then mitosis starts. And the first stage is prophase. During prophase, several important things happen. First of all, notice we don't have what in this picture? What's not there? No nuclear membrane. It goes away. The part that I've got circled here, what's forming there? The spindle fibers, the little ropes, they're forming. Is it what? So, these are the spindles. Also notice that the chromatin is already wound up and it's now in what form? Wound up chromatin we call chromosomes. If we keep going on a little further, by the late part of prophase, the nucleolus now disappears. All the chromosomes eventually hook up to a what? To a spindle fiber. And that's kind of where prophase stops. So the nuclear envelope is gone. The DNA is all wound up and it's all in chromosome form. The spindles are formed. The chromosomes are attached to them. That's prophase. Here's our acronym. Stage 2 is the M stage, the metaphase. And in metaphase, the chromosomes all get pulled to the middle and they get lined up, straight row. That's metaphase, lined up in the middle. Our next part of our acronym is A, anaphase. And in anaphase, those ropes shorten. One set of ropes is pulling this way, one set is pulling this way. And those big egg-shaped bivalent chromosomes get ripped in half. One part of each of those gets pulled toward the top, one part toward the bottom. Again, this part is exactly the same as that part. They're identical. That's anaphase. And then lastly is telophase. And like we said essentially a minute ago, the two sets of chromosomes make it to opposite sides. The DNA then unwinds and goes back to being what? Chromatin. A new nuclear envelope, a membrane, forms around each set of DNA. Spindles go away. And you can notice that the cell is beginning to kind of pinch in the middle. And that's called a cleavage furrow. And it's kind of obvious why it's called that, right? What does cleave mean? What's the word cleave actually mean?
Into, like, crank.
Like you probably have at home a knife called a cleaver. Cleave means to cut.
Into.
Okay? It means to cut. Okay? So it looks like a cut. But that's called the cleavage furrow. That's where mitosis ends. Mitosis ends with only one cell with two what? One cell with two nuclei, identical nuclei. But what always happens right after mitosis? Cytokinesis, which is the division of what? Cell. Okay? Cytokinesis. Mitosis divided the DNA up. Cytokinesis is going to divide the cell up. And that's really already starting before telophase is even over. Cytokinesis is technically not part of mitosis. It's what happens after mitosis. So let's look at the next part. Cytokinesis happens after mitosis. Happens a little bit differently in animals than in plants. What complicates it in plants? A wall, right? That makes it a little more complicated. But in animals, essentially, you start off with, like I said, a cell with two nuclei in it that are the same. But in the middle of that cell is this little ring of proteins. It goes all the way around. And I want you to think of that little ring of proteins sort of like a noose. What can you do to a noose? You can tighten it. So that little noose starts to tighten. And it just keeps tightening and tightening and tightening and tightening until the cell, it may start to look like maybe like this to begin with. And then eventually it just keeps tightening and tightening and tightening and tightening until this side touches this side. What's this made out of? This whole red structure, what's it made out of? What's the cell membrane made out of? Phospholipids. What happens when lipids over here touch lipids over there? They close up, they fuse. It's like two drops of oil touching each other. And eventually what's going to happen is it's going to look like this to begin with. It's going to continue to close and close and close. Eventually, just a minute, I'll come back. Eventually it's going to continue to split off like that until eventually it just breaks into two identical cells. So mitosis divides up the nucleus, the DNA, until eventually, after it's over, cytokinesis splits the cell in half. Let's go back to the slide with the writing on it for a minute. So as I said a second ago, in animals, this cleavage furrow forms due to the contraction of these little proteins. It's almost like a noose. And eventually that noose is going to tighten and tighten and tighten until it pulls the cell into two identical cells. That's how it works in animals. Like I said, in plants it's a little more complicated because you not only got to split the membrane, you've also got to build a new what? You got to build a new wall down the middle. And that's essentially what this is. You get this little thing called a cell plate between the two nuclei, and eventually that forms a new wall, and it also closes up the membrane on both sides of that wall. The main thing to know, though, mitosis divides up the DNA. You end mitosis with one cell with two what? Two nuclei, two identical nuclei. And then right after mitosis is over, you get cytokinesis, which essentially just splits the cell in half, and you end up with two identical cells. You started with one cell, you end up with two cells, just like each other, and just like the cell you started. All right, on the next couple of slides, I've got some pictures. And I'm going to show you the picture, and I want you to tell me from the picture what stage the cell is in. They're not labeled. Cleavage means to cut, split. All right, so this picture right here. Identify the stage of mitosis pictured to the left. Actually, to the right. Should be to the right.
That looks like the.
That's metaphase. Because all the chromosomes have been pulled into the middle. They're all lined up. They're all attached to the spindles. Now they're ready to get broken, right? That's metaphase. Remember, meta means middle. Oops. Got ahead of myself there. All right, what about this one?
That is.
That's prophase. It's kind of probably like right in the middle of prophase. Because the DNA is already wound up, right? We got chromosomes. What's starting to form over here? Spindles. But what hasn't went away yet? Nuclear envelope hadn't went away yet. So that's why it's probably like in the middle of prophase. Nuclear envelope's still there. The new little nucleolus is gone. So most of the things that need to happen in prophase have already happened. Nuclear envelope hadn't went away just yet. And the chromosomes haven't attached up to what yet? The spindles. Spindle fibers. All right, what about this one? That's telophase. Because we still got one cell, but we have two what? Two nuclei. Notice the spindles have already gone away. The nuclear membranes have formed. It's starting to try to split in the middle, but it's not quite there yet. That's telophase. This is telophase right now. Cytokinesis will eventually split it in half. Cytokinesis happens after telophase. All right, thought I had one more picture here. Yes. There's the other one. That's anaphase. Because remember in anaphase, the little ropes shorten, tighten, like that. There were X-shaped chromosomes here. Those got split in half into their chromatids. This set of chromosomes is getting pulled this way. This set's getting pulled that way. This set and this set are exactly like each other. And eventually, cell's going to get split down the middle, and we're going to get two identical cells.
Is that metaphase?
That's anaphase.
Where does the protein go after they cut it?
It's just broken down and reused later. All right, that's mitosis. Now, I'm going to stop talking here for just a minute.